Hello viewers, I am Dr. Robiul. I work as a lecturer of pathology in a medical college hospital and I am making this video for my students and also for you. Hope someone finds this helpful. Today's topic is on lymph nodes. This is the 7th lecture of our ongoing immunology lecture series, and in this video we will learn about introduction and definition of lymph node, and we will also discuss briefly about the fascinating architecture of lymph nodes, and towards the end of today's video we will also talk briefly about lymphocyte recirculation. Okay, a lot of topics, so let's begin. First question, what do we mean by lymph nodes? Always remember, lymph nodes are bean-shaped, encapsulated and vascularized secondary lymphoid organs that are located along the lymphatic channels throughout our body. Lymph node is one of the major anatomic components of our immune system, and as we will see, they have anatomic features that favor the initiation of adaptive immune responses. Human body contains approximately 500 lymph nodes. So, how can we define lymph node? Lymph node can be defined as nodular aggregates of lymphoid tissues that are located along lymphatic channels throughout the body. So I am repeating the definition again for my students. Always remember, lymph node can be defined as nodular aggregates of lymphoid tissues that are located along lymphatic channels throughout the body. So now that we have defined lymph node, now let's move on and talk about the fascinating architecture of lymph nodes. So it is surrounded by a fibrous capsule that extends inside the lymph node to form trabeculae. Beneath the fibrous capsule, there is a sinus system that is called marginal sinus or subcapsular sinus. Incoming afferent lymphatic vessels will pierce the capsule and open into the marginal sinus. So, as you can see, here I have drawn a simple diagrammatic image of lymph nodes using the 3D modeling software Blender. So, special thanks to the Blender team. Now, in this image, we can see a lymph node and it is surrounded by fibrous capsule. Okay, so the arrow is denoting fibrous capsule. Just beneath this fibrous capsule, there is the subcapsular sinus, which is also known as the marginal sinus. And these are the afferent incoming lymphatic vessels. So these are the incoming or afferent lymphatic vessels. And note that these afferent lymphatic vessels are opening into the subcapsular sinus. Now, the subcapsular or marginal sinus will communicate with a delicate intranodal sinus network. Again, this intranodal sinus network will merge into efferent lymph vessels that will exit the lymph node at the hilum. Now, the last two bullet points are very interesting. The endothelial lining of the outer side or subcapsular side of the marginal sinus is similar to that of the afferent and efferent vessels, and it is non-phagocytic. However, the lining of intranodal sinus will have strong phagocytic properties. Let me explain. So in this image, this is the intranodal sinus. This portion is the hilum of the lymph node, and we can see that efferent lymphatic vessel is exiting through this hilum. So this is efferent lymphatic vessel. or outgoing lymphatic vessel, right? But we can also see that in the lining of intranodal sinus there is macrophages and there is also dendritic cells. And that is the thing that was said in the previous slide, that the lining of intranodal sinus has strong phagocytic properties, and they will have such phagocytic property due to the presence of macrophages and dendritic cells in the lining of intranodal sinus. Now, intranodal sinuses are actually lined by reticular cells, and besides reticular cell there are these macrophages and dendritic cells. And intranodal sinuses are lined by reticular cells cross-bridged by collagen fibrils and other extracellular matrix proteins, and they are filled with lymphocytes, dendritic cells and macrophages. A lymph node can be divided into three major concentric regions, and each of these regions will support a distinct microenvironment, as we will see. So, what are the three major regions of a lymph node? This is very important and high yield for your examination. You need to know this. The three major regions of a lymph node will include the cortex, the paracortex and the medulla. So, in this image, this portion of the lymph node is the cortex. Just beneath the cortex, this portion is the paracortex, and beneath the paracortex towards the hilum we can see the medulla. So first, let's talk about the architecture of the cortex of the lymph nodes. It is located beneath the fibrous capsule. It is the compartment where most lymphoid follicles reside. It is composed primarily of B lymphocytes and follicular dendritic cells. So always remember these two names, B lymphocytes and follicular dendritic cells. They are mainly found in the cortex. The appearance of follicle will again depend on their state of activity. And now we will talk about primary follicles and secondary follicles. Primary follicles appear as round aggregates of small lymphocytes containing little cytoplasm. We can also define primary follicles as follicles that do not contain germinal center. On the other hand, secondary follicles will appear after antigenic stimulation, and they will contain pale staining germinal center. So you can remember these two terms, primary follicles and secondary follicles, by remembering that primary follicles won't contain germinal centers, whereas secondary follicles will contain germinal centers. And these are pale staining in the hematoxylin and eosin stained sections, and these germinal centers are again surrounded by a dark mantle zone. So, on hematoxylin and eosin stained sections, germinal center contains pale staining large cells. These cells are highly proliferative and they will show polarization toward the side of antigen stimulation. Germinal center cells are also known as follicular center cells, and they are predominantly B lymphocytes. They will include centroblasts, centrocytes, or small and large cleaved and non-cleaved cells. Germinal center also include macrophages, follicular dendritic cells, and a population of T follicular helper cells. Now let's talk about the mantle zone. it surrounds the pale staining germinal center, and mantle zone B lymphocytes are small. They contain little cytoplasm and they stain dark blue with hematoxylin and eosin stain. Proliferated germinal centers will indicate humoral antibody production. So in this image, these are the lymphoid follicles. So now that we have talked about the architecture of the cortex, now let's talk about the architecture of the paracortex of the lymph nodes. Always remember, it is the zone that is situated between the cortex and the medulla. It contains the mobile pool of T lymphocytes that are responsible for cell-mediated immune responses. Paracortex also contains post-capillary venules, which are lined by specialized high endothelial cells. These post-capillary venules are also called high endothelial venules. And also remember that paracortex also contains interdigitating dendritic cells. Now, we have just mentioned an interesting term, that is high endothelial venules, or HEV. So what do we mean by this term? So in this image, this portion is the cortex. We can also see the paracortex, the fibrous capsule and subcapsular sinus. And in the paracortex region, we can see high endothelial venules and T lymphocytes. So let's talk about high endothelial venules now. They are specialized blood vessels that are adapted for lymphocyte trafficking. They are formed by specialized endothelial cells that are unusually tall as opposed to the usual thinner endothelial cells of regular venules. Since they are unusually taller than the usual thin endothelial cells of regular venules, that's why they are called high endothelial cells. And the venules are therefore called high endothelial venules, or HEV. In humans, high endothelial venules are found in lymph nodes, tonsils and adenoids in the pharynx, Pierce patches in the small intestine, appendix, small aggregates of lymphoid tissue in stomach and large intestine. Okay, so these are the sites where we can find high endothelial venules. Now, naive lymphocytes will enter the lymph node by passing between these specialized endothelial cells of high endothelial venules. These cells express specialized ligands for lymphocytes and can support high level of lymphocyte extravasation. They express addresins. Addresins are specific adhesion molecules that can attach to L-selectins on lymphocytes and anchor them to the high endothelial venule wall in preparation for crossing the endothelium. So in this image, this is the high endothelial venule, and note that the high endothelial cells that are lining this high endothelial venule, they are quite plump and tall looking. So that's why the cells are called high endothelial cells and the venule is called high endothelial venule. Also, we can see in this image the fibroblast reticular cell conduit and the fibroblast reticular cells that are lining this conduit. So now let's talk about these new terms. What do we mean by fibroblast reticular cells and what do we mean by fibroblast reticular cell conduit? Now always remember the paracortex is also traversed by a web or network of processes that arise from stromal cells called fibroblast reticular cells. So we can see that these fibroblast reticular cells are in fact the stromal cells of the lymph nodes, and they are forming a network of processes, and that network is known as fibroblast reticular cell conduit system. The conduits range in diameter from 0.2 to 3 micrometer, and they contain organized arrays of extracellular matrix molecules, collagen fibers, fibrillin microfibers, etc. that are surrounded by a basement membrane that is produced by sleeve of fibroblast reticular cells. Now, you may be asking me, Dr. Rabiul, what is the role of such fibroblast reticular cell conduit system inside our lymph nodes? Why do we need them? And that's a very good question, and in order to understand the role of such conduit system, we have to know about a fascinating process that is happening inside our lymph nodes. Always remember, the lymph nodes have an innovative size-dependent sorting process for initiation of antigen processing. And this sorting process also depends on the microanatomic features of the lymph nodes. So when dendritic cells with phagocytized antigens are coming to the lymph node, they can enter the regional lymph nodes via afferent lymphatic vessels. Okay, so these dendritic cells, they were present in some peripheral tissues, and there they had encountered certain antigens, and then they phagocytized those antigens, and now they want to show those antigens to the T cells in the lymph node. So what are they doing? They are going to the lymph node, and they can enter the regional lymph nodes via afferent lymphatic vessels, and then they are migrating to the T cell zone of the lymph node due to the chemotactic stimuli, and there in the T cell zone, there the antigens are being processed and presented. So what is happening when we are talking about soluble lymph-borne substances? Always remember, soluble lymph-borne substances like intact viruses and high molecular weight particles also enter the lymph nodes via afferent lymphatic vessels. And when they go in the subcapsular sinus, particularly when they are inside the intranodal sinus, they are engulfed by macrophages or dendritic cells, and then those macrophages and dendritic cells will process and present the antigen to the cortical B lymphocytes. Recall that in the first half of this video we had talked about the lining of the sinuses that we see inside the lymph node, and the lining of the intranodal sinus had phagocytic properties. We had already talked about that, and that phagocytic property of the intranodal sinus was due to the presence of macrophages and dendritic cells in their lining besides the reticular cells. But what will happen when lower molecular weight molecules have entered the subcapsular sinus of the lymph nodes? Since they cannot penetrate the floor of the lymph node sinus, so instead they will flow via the tubular structures that we are calling fibroblast reticular cell conduits. Always remember, dendritic cells are located very close to these conduits, and their processes are intercalated between the fibroblast reticular cells that are lining these conduits. So the dendritic cells can take up and process the lower molecular weight molecules from these conduits, and then they can present the antigens to the T lymphocytes. So that is the role of fibroblast reticular cell conduits inside our lymph nodes. So in this image, we can see the high endothelial venule. We can also see fibroblast reticular cell conduit, and we can also see that the conduit is lined by fibroblast reticular cells. And we can also see a dendritic cell, and if we look carefully, we can see that the process of this dendritic cell is intercalated between two fibroblast reticular cells that are lining this fibroblast reticular cell conduit. So this is the dendritic cell process that is intercalated between two fibroblast reticular cells. So now that we have talked about the architecture of paracortex, now let's move on and talk about the architecture of medulla. It is located close to the hilum. We have already seen that when we were describing the image of the lymph node, and it grows in the form of cords. Medulla is rich in lymph sinuses, arteries and veins. However, it contains minor amount of lymphocytes, which are a mix of T lymphocytes and B lymphocytes. Now the last topic that we will talk about is regarding lymphocyte recirculation. So let's talk about that now. Lymphocytes are constantly recirculating between blood and lymphoid organ, and this is very important for immune surveillance. As we can see in the second bullet point, successful immune surveillance will depend on such endless patrolling of lymphoid tissues by lymphocytes that are searching for foreign antigens. And as we will see, this is particularly important for T lymphocytes. Now always remember, lymphocytes can enter the lymphoid tissues either through blood vessels or through afferent lymphatic vessels. For example, most of the lymphocytes will enter the lymph nodes through blood vessels, and if I want to be more specific, they will enter with the help of high endothelial venules. The remaining 15 to 20% of the lymphocytes, along with dendritic cells and antigens, can enter the lymph nodes through afferent lymphatic vessels. In the high endothelial venules, lymphocytes will interact with the vessel wall in multiple steps. To leave the high endothelial venules, the blood-borne lymphocytes will first tether to the high endothelial cells that are lining the high endothelial venules, and then the lymphocytes will start to roll. After an activation step, they will firmly adhere to the vessel wall. Now always remember, high endothelial venules can express specialized ligands for lymphocytes, and it can support high level of lymphocyte extravasation. High endothelial venules can express addressins. What are addressins? These are specific adhesion molecules that can attach to the L-selectins on the lymphocytes and anchor them to the high endothelial venule wall in preparation for crossing the endothelium. Then the lymphocytes will transmigrate through the vessel wall and navigate through the stroma of the lymph node following chemotactic guidance. Now, naive B lymphocytes express chemokine receptor CXCR5, and naive T cells express chemokine receptor CCR7. Always remember, chemokine receptor CXCR5 tends to bind specifically to chemokine CXCL13 that is produced by follicular dendritic cells. On the other hand, the chemokine receptor CCR7 that is expressed by naive T cells, that chemokine receptor tends to bind specifically to chemokine CCL19 and CCL21 that are produced by stromal and dendritic cells of the T cell region in the paracortex. So we can see that due to interaction between the chemokine receptors and the chemokines, B lymphocytes will tend to migrate to the follicles of the lymph node, and T lymphocytes will tend to migrate to the T cell region following the chemokine gradient. Now, this anatomic segregation of B and T cells in the lymph node will ensure that each lymphocyte population is in close contact with the appropriate antigen presenting cells. B and T cell populations are kept apart until it is time for them to interact in a functional way. Naive T cells are activated by antigen and co-stimulators in the peripheral lymphoid organs, and activated T cells will proliferate and differentiate into effector cells that will migrate to the site where the antigen is present. After the infection has resolved, most of these activated T cells will die by apoptosis, and few may persist as memory T cells. Now, I have an entire video on apoptosis, so you can also watch that video after finishing today's video to know more about apoptosis. Naive B cells are also activated by antigen and co-stimulators in the peripheral lymphoid organs, and activated B cells can either differentiate into plasma cells that will secrete antibodies specific for the antigen, or they can differentiate into memory B cells and stay in the follicles waiting to respond to reinfection. So this was in short about lymphocyte recirculation. So this concludes our lecture on lymph nodes. I hope this video was helpful. If you like my videos, do comment, share, subscribe, and let me know. And for my students, I will also recommend you to go through your textbooks to know more. Okay, that's all for today. Until next time, take care and stay blessed. Thank you.